subjects. So we need really high numbers of individuals to use in that type of methodology, and we're talking about thousands. So we use those that have known genotypes and phenotypes. So we measure each one in M genetic locations. So they represent common variation in the whole genome. So typically we're looking at so-called SNPs, or single nucleotide polymorphisms. So those are point mutations that are informative. There's some variation in populations because if there is no variation, if there's no variation in our species or in a population, of course, such position cannot be used as a marker because it's not informative. So we need genetic variation to correlate it with phenotype, and we need large numbers of those as well, of these genetic markers, and it's between hundreds, thousands to a million or more. Recently, we're moving to whole genome sequencing, so we're using all nucleotides within the genome, but which is like more than 3 billion nucleotides. Realistically, we don't really gain much information compared with informative SNPs, because most of these sites, most of the nucleotides in the genome are not variable. So even it's good to have all genome information, but again, when we talk about GWAS, we don't really gain much compared with a collection of SNPs, because, like I said, we need genetic variation to run the mapping. Now we can think of our data as matrix, with subject as rows and SNPs as columns. So X will be the genotype and Y will be the phenotype. So our first task is to do this association testing. And so we need to find SNPs or genotypes, which is the columns, will be organized into columns, X, that are statistically associated with Y. And then we need to run statistical tests, which can be think of as a test, like M separate statistical test. So M refers back to the number of genetic markers or genotypes, and it maybe will be more clear when I show you how this works. So let's assume that we have two phenotypes, cases, or the phenotype of interest and controls, and we get the SNP information or sequence information from each individual. But the whole study is divided into two groups. So one is phenotype, the other is control, no phenotype. And those in red are SNPs. Those in red are positions that differ for the mutation. Yes. So Xij refers to a specific position within a specific marker within the matrix. So this could be a position like this. Okay. So again, those in red are those that are variable, and which we call single nucleotide polymorphisms. The rest of them is not variable. so it's not very informative. And we want to find, again, association between the variation within each locus or the genotype and the phenotype. So we can construct, and of course, we need to find, we expect that those that are most differential, that let's say, ideally unique per group. So let's say that G is unique or almost unique to the phenotype, to the disease phenotype, and T occurs in the negative control. So it's something that we want to identify because this is what suggests some statistical association, right? So if, let's say that we have 500 individuals, we have 500 rows within cases and 500 controls for comparison, not just whatever the number is less than 10. We're talking about hundreds per group, right? And the goal is to find a marker that differentiates, that separates these two groups. Ideally, again, we want a marker that that it's really perfect separation, which is, in other words, unique or specific per group. But as you can imagine, this is quite rare. Typically, what we see something like that. We see tendency, right? That let's say here is 1, 2, 3, 4, 5, 6, 7 T's, but there's one G in control, and the other way around in the disease phenotype, so there's one T and 7 G's. So is this significant association or it's not? This is the question. That's why we're applying statistics to give us numbers, to give us some probabilities associated with these associations. So basically, what it means is that based on every single SNP like that, we construct a table, and this could be a 2x2 table that you are well familiar with now, or it could be 2x3, because we may have 3 genotypes. We may have heterozygotes and 2 types of homozygotes. If you want to think of SNPs like before, you can think of like A, G, and the third one, the genotype second would be something else, right? Because it's possible that other nucleotides can be there. And then we have 2 phenotypes, healthy and sick, and we're testing for statistical association. So the problem here to test is, well, we need to formulate the hypothesis to test. So the null hypothesis from statistics is that there is no connection between disease and SNP, and we want to reject the null hypothesis using some statistical tests like chi-square. So in this example, when we have 2x3, we would use probably chi-square test. If it's 2x2, we would use like Fisher's exact test. But you don't need to worry about the exact statistical test we need. I'm just showing you this as to give you a flavor what this GWAS is about. So, like behind every single marker, we have a table and statistical test. So again, where is the table from? The table is based on counts from pictures like that. Of course, everything is automatic. It's we have computer programs to do it for us, so we don't go over every single position and count them manually. But this is the principle basically, that we have 2 phenotypes. So this is phenotype 1, phenotype 2. Let's say this is disease, this is control or healthy, and we have 2 markers, or we may have more than 2 genotypes. We have 3 genotypes. So every based on every column like that, we construct a table and a statistical test, right? So what's next? What do we do next with it? So we need to map it against the genome. Now we know every position, we know where it is located, we know its geography, in other words. Because this is sequencing information, we map it back to the chromosome. So we know chromosomal position of every position like that. So we construct a map like this, in which every single dot represents a statistical test, and a table like that I showed you before. So the horizontal axis is for the genetic position, right? And again, every dot is a statistical test from a table based on the counts that I just showed you. And this is the position is plotted against statistical significance from the test. So here there's some negative log of the p-value. Regardless what it is, think of it as statistical significance, and the higher value on the vertical axis, the higher statistical significance, or the tighter statistical association between phenotype and the genotype, the marker. So those points that are high represent tests that show us some significant or at least interesting association between some genetic markers and the phenotype of interest. Now, we need to use some statistical criteria. Those of you who do statistics remember that the most commonly used threshold for statistical significance for the p-value is 0.05, right? This is how if a test produces p-value 0.05 or less, then we take it as a significant result. So this level of statistical significance 0.05 corresponds to this black line, which means that every dot above this black line is statistically significant at this level, at this threshold of 0.05, which is thousands, and we cannot really make much sense out of it, right? And the problem is that this level tells us about the statistical significance obtained by chance alone. So it can be a false positive. So when we have 100 statistical tests and we reject the test 100 times, we expect 5 times to reject it incorrectly. All right, this is what the statistical threshold tells us. So 5 rejections will be false positives. When we have 1,000,000 tests, because we have 1,000,000 markers, and for every marker we run the test. Now we multiply 1,000,000 times 0.05, which we expect 50,000 false discoveries. So, like most of them above the black line are false positives, which is expected by statistics alone. So what's the solution? The solution is to be very selective in what results we declare as significant. So we can make the criteria, the statistical significance criteria, more stringent, and the current p-value that we accept as significant is 5 times 10 to the negative power of 8. So if we actually for this one, we don't need it so stringent. We can make it 10 to negative 5, which is the red line, and then we're looking at points that are located above the red line, and we find only one of them. So we take this, I don't know if you can see it, this blue dot as a significant result. So that will be the gene or the genetic marker that is associated statistically with the phenotype of interest. So again, we declare only one association, and we can actually, we know where the gene is. So it turns out that this is chromosome 7, and we specifically know where this position is. This is an example of results of a large GWAS study dedicated to 7 disorders, including bipolar disease, coronary artery disease, Crohn's, hypertension, and so on. There were 14,000 cases analyzed, compared with 3,000 shared controls, and the cost was huge. I mean, it was a large experiment. It resulted in only 13 significant findings at the stringent level of 5 times 10 to negative 8. So you can see those as these green spikes here for these disorders. So this is how we declare that, okay, we found we found a gene for Crohn's disease. So the gene is somewhere here on chromosome 5, and we actually have this information. We can go back. The computer finds it for us, so we can tell where the marker was. Okay, but we start with a huge number. We start with like 1 million markers, and then we narrow down those that correlate with the phenotype, which is shown here. Does it make sense? We talked about heritability, and I know it's a difficult concept, but turns out that, so the hope was that using GWAS, we can actually explain heritability. That because GWAS gives us ultimate information about the genetic variants and the phenotype. So in other words, it should show us what this heritability is. But turns out that that GWAS, for some reason, does not explain heritability. And for example, for height, from twins and a family study, we know that the heritability is 80%. So it's high, but from huge GWAS studies that involved more than 40,000 people or subjects, GWAS explained only 10% of the height variability. Same with schizophrenia, heart disease, cancers. The heritability is high, between 30 and 80% for those, but GWAS accounts for only between 5 and 10%. So basically, for all complex traits investigated, there is a major gap, and there is some disappointment related to GWAS. So where is all the heritability? So this is the case of the missing heritability, and there are some speculations. The first one is that not all rare variants are covered by GWAS. Every family has its own mutation. And we know examples like BRCA, that this is the case. Or there could be some complex associations, some interactions between SNPs that actually, rather than the genotype itself, it's actually interaction between the genotypes that determines the phenotype. And so we call it epistatic interaction. So it's like interaction, some functional interaction between independent loci or independent genes. And the problem is that when we have 1 million markers, million SNPs, and we start considering now interactions between them, we have 10 to 12 possible pairs, which is not possible to analyze at this point. The alternative is that we lack power, that the effects are weak, and we still need more data than currently we can accumulate. Another explanation is that maybe there are some epigenetic effects that, which means that epigenetic is something that is not it's not that we see at the genetic marker level, right? By definition, epigenetic is inheritance unrelated to genetic mutations, and to some extent, these speculations have been tested. But the bottom line is that we still don't know what's going on with GWAS, why GWAS underestimates heritability. Okay, we get to move on because the next one